So what actually fuels you? And I don't just mean your morning coffee, right? I mean the incredibly complex machinery that's humming away inside every single cell in your body nonstop. Today, we're taking a deep dive into that really fundamental question: how our cells get, transform, and, well, use energy. Welcome to the deep dive.
Yeah, welcome. It really is a mission, you could say, to get to the core of life itself. We're going to explore the absolutely central roles of um, adenosine triphosphate ATP, that's the universal energy currency we always hear about,
Right.
and oxygen, O2, empowering pretty much all biological activity. We'll try to uncover the sophisticated mechanisms that keep our bodies running, look at what happens when these systems falter, which they do.
Inevitably.
Inevitably, yeah, and sort of bridge these fundamental biochemical processes to the daily experiences of health and disease.
And we promise a journey that's clear, accessible, but also genuinely insightful, we hope. We'll guide you step by step through the key concepts, the crucial pathways, and bring in some real-world clinical examples that show exactly why this stuff matters. You'll gain, hopefully, a much deeper understanding of how every breath you take, every move you make, right down to that cellular level, is this act of, like, a finely tuned energy management.
Okay, so if our cells are constantly working, what's the fundamental power source? The currency they all seem to spend for everything. You mentioned it, ATP.
Exactly. ATP, it's not just some molecule, it's really the heartbeat of cellular energy. It's constantly cycling between its high energy form, triphosphate, and its lower energy form, ADP diphosphate. And the beauty of this ATP ADP cycle is its continuous regeneration. We generate ATP by essentially breaking down fuels, I think carbohydrates, fats.
The food we eat. Precisely. Through a series of oxidative reactions, the chemical bond energy from these fuels gets transferred to these specialized coenzymes, like NAD plus and FAD. They get reduced to NADH and FAD2H. You can think of these reduced coenzymes as tiny rechargeable power banks loaded up with high-energy electrons.
Okay, so these little power banks get charged up from our food. How do they then, deliver that energy to actually make the ATP?
Right, that's where the recharge part really comes alive. These electron-rich coenzymes, NADH and FAD2H, they travel to what's called the electron transport chain, or ETC. There, they get oxidized, they give up their electrons ultimately to oxygen. As electrons pass down this chain of protein complexes, the energy released is precisely harnessed. It's used to pump protons, create this electrochemical gradient across a membrane.
Like building up pressure behind a dam.
Sort of, yeah, that's a decent analogy.
Yeah.
And that pressure, that gradient, then drives an enzyme called ATP synthase to regenerate ATP from ADP and inorganic phosphate. This whole process, which is highly dependent on oxygen, that's what we call oxidative phosphorylation, it's really elegant, this conversion of electron potential into usable energy currency.
And once we have that freshly minted ATP, our cells immediately start spending it, right? The energy stored in those high-energy phosphate bonds, that powers nearly all cellular work. It's actually kind of astonishing, the variety of tasks this one molecule enables.
It really is. ATP powers three main types of work. First, there's mechanical work, picture muscle contraction. It's like this perfectly choreographed molecular dance. ATP is the fuel directly powering that movement. It literally changes the shape of proteins.
How does it do that?
Well, for instance, in your muscle fibers, when ATP is hydrolyzed, broken down, the energy released basically cocks the myosin protein head that allows it to bind to another protein, actin, and pull, generating force. And consider this. An exercising muscle can need almost 100 times more ATP than a resting one.
Wow. That's a huge difference.
It is. And ATP also drives other molecular motors, things like kinesins. They act like tiny trucks, hauling vital cargo vesicles, organelles along these cellular highways, the cytoskeleton.
So movement isn't just muscles flexing, it's happening constantly, deep inside every cell.
Okay.
And then there's transport work.
Yes, absolutely crucial for keeping the cell's internal environment stable, maintaining equilibrium. Think of the sodium-potassium ATPase, sometimes called the NAL plus K plus ATPase or sodium-potassium pump. It works tirelessly in basically every cell membrane.
I remember hearing about that one.
Yeah, it's fundamental. This pump uses ATP energy to actively move ions specifically. It pushes sodium out of the cell and pulls potassium in against their natural concentration gradients. It's like pushing water uphill, energetically speaking. This constant pumping maintains crucial cellular balance, it sets up electrical gradients needed for nerve impulses, and it even helps control cell volume. And what's really remarkable is that this active transport alone accounts for a huge chunk of your basal metabolic rate, your BMR.
So even when I'm just sitting here, or sleeping.
Exactly. Even when you're fast asleep, a significant portion of your energy expenditure is dedicated just to keeping these pumps running, maintaining this fundamental balance.
Okay, mechanical work, transport work. What was the third one?
The third is the vast amount of biochemical work that ATP enables. This is where ATP fuels what we call anabolic pathways, basically, building larger, more complex molecules from smaller pieces. Think about synthesizing DNA for cell division, or building proteins from amino acids, or storing glucose as glycogen for later use.
Building things takes energy.
It does. These are all energy-requiring processes, and ATP provides that essential energy investment. It also powers critical detoxification reactions. For example, your liver converts toxic ammonia, a byproduct of protein breakdown, into urea, which is much safer to excrete. That takes ATP. All these complex biological constructions and transformations rely entirely on ATP. And interestingly, any energy from those initial fuels that isn't ultimately captured and used for one of these types of work, it's just released as heat.
Which helps keep us warm, I suppose.
It does contribute to body temperature, yes.
So it's this constant churn of ATP used up, then regenerated. But you mentioned balance. Our bodies are masters of balance, aren't they?
They really are. The body's ability to maintain ATP homeostasis is, well, phenomenal. Regardless of whether you're sprinting flat out or just resting quietly, your cells manage to keep ATP levels incredibly constant. They do this by precisely regulating the rate of fuel oxidation to match the demand. What's truly remarkable isn't just that ATP levels are maintained, but the sheer speed and precision with which our bodies can switch fuel sources, burn more fat, less sugar, or vice versa, and adjust production on a dime. It's a testament to billions of years of evolution.
But that balance can be disrupted.
Oh, absolutely. When this delicate balance is thrown off, we see profound clinical issues. Think about conditions like obesity or hyperthyroidism, or the severe immediate impact of a myocardial infarction, a heart attack, all fundamentally linked to energy metabolism going wrong.
Okay, we've seen what ATP does and broadly how it's generated. But behind all these transformations, there must be some fundamental rules of energy, right? Some physics involved. Let's maybe pull back the curtain on that a bit. You mentioned Gibbs-free energy.
Right, Gibbs-free energy, or Gibbs, Kurji. It's a concept from thermodynamics, but it's essential for understanding biochemistry. Guy basically tells us the maximum amount of useful energy that can be obtained from a biochemical reaction occurring at constant temperature and pressure. It helps predict if a reaction will proceed spontaneously without needing an external energy push. Now, we often talk about G note, delta G naught prime. That's the energy change under very specific standardized conditions. pH 7.0, 25 degrees Celsius, and 1 molar concentrations of everything. It's a useful reference point.
But cells aren't usually at 1 molar concentrations of everything, are they?
Almost never. And that's a key insight. The actual gauge inside a living cell can be very different from the standard DG to 1 low. Why? Because the actual concentrations of reactants and products in the cell are constantly fluctuating and are rarely, if ever, at that standard 1M level. The cell's dynamic environment constantly shifts the actual favorability of reactions.
Okay, so some reactions just happen, they release energy, and others need energy put in.
Exactly. Reactions that release energy are called exergonic. They have a negative E and usually a negative 0. Their products are more stable, meaning they have lower chemical bond energy than the reactants started with. Fuel oxidation, breaking down molecules like glucose or fats for energy, that's a prime example of exergonic processes. Conversely, endergonic reactions require an input of energy. They have a positive E and usually a positive 0G. Their products are less stable, higher energy. ATP synthesis itself, making ATP from ADP and phosphate, is a classic endergonic reaction. It needs energy input.
So how does life keep going if some essential steps need energy?
Well, for life to continue, the overall metabolic pathways in our cells must have a negative E. They must be energetically favorable overall, even if individual steps within the pathway are endergonic.
Right. And this is where ATP's famous high-energy phosphate bonds come in. What is it about ATP's structure that makes it such an effective energy currency? Why high energy?
It's quite elegant structurally. The bonds linking the phosphate groups in ATP, especially the last two, are often called high energy, but maybe unstable is a better way to think about it. This instability comes from the cluster of negative charges on those phosphate groups. They repel each other, creating the sort of molecular tension like a compressed spring.
Okay, like charges repel.
Exactly. So when one of these bonds is broken, usually the terminal one, through hydrolysis, adding water, that tension is released, and it releases a significant usable amount of energy. Under standard conditions, DGOBS, it's about negative 7.3 kilocalories per mole. That's a substantial packet of energy in cellular terms. And this specific amount isn't random, it's kind of perfectly tuned. Too little energy release wouldn't be enough to power many essential reactions. Too much could be wasteful or even damaging. It's really a cornerstone of efficient energy transfer in biology.
So cells need to do things that cost energy, these endergonic processes like building molecules. How do they pay for it using ATP?
That's where the genius of coupling comes in. The body achieves these energetically unfavorable processes by directly linking them, chemically linking them, with an energetically favorable reaction most often, the hydrolysis of ATP. Let's take an example. Synthesizing glucose 6-phosphate, which is the very first step in storing glucose as glycogen, is actually endergonic on its own. It wouldn't happen spontaneously.
Okay.
But the cell couples this reaction with the cleavage of an ATP molecule. The energy released from breaking that ATP bond is used to drive the formation of glucose 6-phosphate. Because the Ag values for a sequence of coupled reactions are additive, the large negative Ag from ATP hydrolysis effectively pays for the positive Ag of the glucose phosphorylation.
Yeah.
This makes the overall coupled reaction energetically favorable, negative beta, allowing the entire process to move forward.
So it uses the energy from one reaction to immediately power another one. That sounds incredibly efficient.
It truly is. And remember, while Ag gives us a baseline, the actual deed in the cell is highly dependent on those local substrate and product concentrations. This means a reaction that might look unfavorable under standard conditions. Positive Ag can actually be pulled forward in the cell. How? Well, if, for instance, the product of that reaction is immediately snatched up and used by the next enzyme in a pathway.
Keeping its concentration low.
Exactly. Keeping the product concentration low can make the actual D negative even if Ag was positive. Or if the substrate concentration is kept very high. This dynamic flexibility ensures that cellular processes can adapt and keep flowing, constantly optimizing energy transfer based on the cell's needs.
Okay, we've talked a lot about ATP as the currency, but where do the paychecks, the energy that ultimately gets converted into ATP, actually come from? Let's dive into the source of that power.
Right, that takes us back to fuel oxidation. Fundamentally, oxidizing our fuel, breaking down carbohydrates, fats, proteins, is all about transferring electrons.
Yep.
Those reduced coenzymes we mentioned earlier, NADH and FAD₂, they are essentially those paychecks. They are electron carriers. They capture high-energy electrons harvested from our food.
During breakdown.
During breakdown, yes. And they transport these electrons to the electron transport chain. That's where the energy carried by those electrons is ultimately used to generate the bulk of our ATP. So when a fuel molecule is oxidized, it loses electrons. When NAD plus or FAD accept those electrons, they become reduced to NADH or FAD₂. It's this constant back and forth of oxidation and reduction redox reactions.
There's that LEO-GER acronym sometimes used, right? Loss of electrons is oxidation, gain of electrons is reduction.
That's the one. It's a handy way to remember it. And NAD plus and FAD don't accept electrons in exactly the same way, do they? They have slightly different chemical jobs.
You mentioned they have different specialties.
They do. NAD plus typically accepts 2 electrons and a proton, effectively packaged as a hydride ion. It's often involved in reactions, oxidizing alcohols or aldehydes. FAD, on the other hand, accepts 2 electrons as 2 separate hydrogen atoms. It's frequently involved in reactions that form carbon-carbon double bonds. This chemical specificity allows different enzymes to utilize the right coenzyme for the specific type of redox chemistry they need to perform.
And the ultimate destination for all these electrons being passed around in aerobic respiration.
Oxygen, molecular oxygen, O2. We can quantify a compound's tendency or willingness to accept electrons using something called its reduction potential, or EC/NO. The more positive the reduction potential, the greater its affinity for electrons. Oxygen has the highest positive reduction potential among common biological molecules.
So it really wants those electrons.
It really wants them. This makes oxygen the ultimate electron acceptor at the very end of the electron transport chain. Its strong pull on the electrons is what drives the entire flow down the chain, efficiently releasing the energy that's used for ATP synthesis. It's literally why we need to breathe every few seconds to supply that final electron acceptor.
Okay, so this connects to something practical. Why fats have more calories than carbs, right? It's about how reduced they are.
Exactly. This links directly to a fuel's caloric value, how much energy we can get from it. Highly reduced fuels, like fatty acids, are packed with carbon-hydrogen-CH bonds. They have lots of electrons to donate during oxidation. For instance, palmitate, a common fatty acid, yields roughly 9 kilocalories per gram when fully oxidized. Partially oxidized fuels, like glucose, already have quite a few bonds with oxygen, CO, OH. They have fewer high-energy electrons left to donate, so they provide less energy, about 4 kilocalories per gram.
And alcohol, ethanol.
Ethanol is somewhere in between, about 7 kilocalories per gram. It's more reduced than glucose, but less reduced than fat. And it's also a crucial point that not everything we might consume has caloric value for humans. We can't get energy from eating wood, for example.
Thankfully.
Ah, yes, because we lack the enzymes needed to break down and oxidize cellulose, the main component of wood. Same goes for something like cholesterol. Although it's a lipid, its complex ring structure can't be readily broken down by our metabolic pathways to yield energy.
That makes sense. Now, speaking of similar molecules, what about NADPH? It sounds almost identical to NADH. Is it just another electron carrier for ATP production?
You've hit on a really critical distinction. NADPH is structurally very similar to NADH.
Mm-hmm.
It also carries high-energy electrons, and it has basically the same reduction potential. But it has an extra phosphate group attached to its ribose sugar component. This seemingly small difference acts like a label. It drastically changes how enzymes interact with it.
So enzymes can tell them apart.
Precisely. And as a result, while NADH is primarily funneled into the electron transport chain, specifically for ATP generation, NADPH is predominantly used directly in a different set of reactions. Crucial biosynthetic reactions, analysis, like making fatty acids or cholesterol. It's also vital for detoxification processes, particularly in protecting cells against damage from reactive oxygen species, oxidative stress. It donates its electrons for these building and protective jobs rather than primarily for ATP.
Okay, different roles despite the similar names. Good to know.
And we absolutely cannot forget anaerobic glycolysis. We focus heavily on oxygen-dependent energy production.
Because it's so efficient.
Immensely efficient, yes. But anaerobic glycolysis offers a vital backup plan. It's a pathway that can generate a small amount of ATP without requiring oxygen. Now, it's much less efficient than oxidative phosphorylation. You get far less ATP per molecule of glucose, but it's absolutely crucial in certain situations. Think about cells with limited or no oxygen supply, like mature red blood cells, which don't even have mitochondria. Or consider your skeletal muscles during an intense sprint when oxygen delivery can't keep up with demand.
They switch to anaerobic.
They rely heavily on it for that quick burst of ATP. And critically, it's important for tissues like the heart during a myocardial infarction when blood flow, and therefore oxygen supply, is severely restricted. There's a short-term fix, but it can be a lifesaver buying the cell some time.
It really highlights how adaptable our energy systems are. Okay, it's one thing to understand all this biochemistry in theory, but how do these principles actually play out? How do they manifest in real-life health scenarios? Let's try and bridge the science to the clinic now.
Absolutely. Let's start with Otto S. He's a medical student who, like many students under pressure, experienced significant weight gain during his studies. Otto's case is a really vivid illustration of that fundamental principle of energy balance. When caloric intake consistently exceeds energy expenditure.
Calories in versus calories out.
Basically, yes. The expenditure includes his basal metabolic rate, the energy needed just to keep basic functions running, plus any physical activity. When intake is higher than output day after day, the excess energy has to go somewhere. The body, being geared for survival, efficiently stores that surplus energy, primarily as fat in adipose tissue. Otto's journey, gaining nearly 60 pounds from 154 pounds to 210 pounds, despite theoretically knowing better as a med student, just underscores how easily even slight, persistent imbalances between diet and exercise can impact our metabolic health and lead to obesity.
Right, so that's a case of energy storage getting out of hand. What about the opposite problem? What if the body starts burning through energy too quickly? That brings us to Stanley T, a 26-year-old man presenting with symptoms like feeling hot all the time, losing weight even though he's eating well, a racing heart, and feeling nervous.
Stanley's symptoms paint a classic picture of hyperthyroidism. This is a condition where the thyroid gland is overactive and secretes excessive amounts of thyroid hormones, mainly T3 and T4.
Now, these hormones act like accelerators for many of our basal metabolic processes. They significantly increase the rate of ATP use by various cellular processes.
Like those pumps we talked about.
Yeah.
Exactly, particularly the Na K ATPase. Its activity is ramped up, increasing transport work. This increased ATP consumption naturally demands more ATP production, which means more fuel oxidation. This skyrockets his basal metabolic rate, BMR. Burning all that extra fuel generates a lot more heat, explaining his heat intolerance and sweating. Thyroid hormones can also, in excess, make ATP production slightly less efficient, meaning the mitochondria burn fuel, but generate more heat relative to the ATP produced. It's like an engine running inefficiently hot.
So he's burning through calories like crazy.
Precisely. This dramatically increased energy expenditure leads to weight loss, even though he might be eating more than usual, because his body is simply consuming energy reserves far too rapidly. And the increased activity of the sympathetic nervous system, which is also influenced by thyroid hormones, contributes to his rapid heart rate and feelings of nervousness or anxiety.
Okay, we've seen imbalances leading to storage and excesses leading to overexpenditure. But what about a sudden catastrophic energy failure? Something that really pushes cells to their absolute limit. That sounds like the situation with Cora N and a myocardial infarction, a heart attack.
Yes, Cora's heart attack represents a severe, acute disruption of ATP production. The underlying cause is usually hypoxia, a critical lack of oxygen reaching a part of our heart muscle. Remember, the heart muscle is incredibly energy hungry. It relies almost entirely on oxidative phosphorylation to make enough ATP to sustain its constant contractions. Each heartbeat uses a significant fraction, maybe around 2% of the cell's ATP,
So it means a constant supply.
a huge constant supply. Without oxygen, oxidative phosphorylation, the main ATP generator, grinds to a halt. This leads to a rapid and catastrophic drop in cellular ATP levels. This ATP deficiency quickly cripples those energy-dependent active transport pumps we discussed, especially the Na plus K plus ATPase and also the T2 plus ATPases, which pump calcium out of the cell or into storage.
What happens when those pumps fail?
They can no longer maintain the normal, steep ion gradients across the cell membrane. Sodium starts leaking in and isn't pumped out. Potassium leaks out. Critically, calcium levels inside the cell begin to rise uncontrollably. This influx of sodium and calcium, along with other ions like hydrogen ions from increased anaerobic glycolysis, causes the cell to swell due to osmotic effects. More importantly, the high intracellular calcium activates damaging enzymes and triggers various cascades that lead to inflammation and ultimately necrosis, uncontrolled, messy cell death.
And that's the heart muscle damage seen in a heart attack?
That's the core of the damage, yes. Doctors can actually measure proteins like troponin, which leak out of these dying heart cells in the blood to help diagnose a heart attack. The body does try to adapt, activating things called hypoxia-inducible factors, HIFs. These are proteins that turn on genes trying to help cells survive under low oxygen conditions, promoting anaerobic glycolysis and new blood vessel growth. But often, during a severe heart attack, these responses aren't fast enough or sufficient to prevent widespread cell death in the affected area.
It really drives home how central and critical oxygen and ATP production are, second by second. So let's recap. We've taken quite a deep dive today into cellular bioenergetics. We've unraveled how ATP acts as that essential energy currency for the cell. We explored the thermodynamic roles, like Gibbs free energy, that govern these energy transformations, looked at the different kinds of cellular work ATP powers, mechanical, transport, biochemical, and how our bodies generate and regulate this vital energy molecule.
Yeah, and we've really seen just how tightly linked ATP production is to oxygen, haven't we? And how even seemingly subtle shifts in energy balance, like in Otto's case, or major hormonal influences like in Stanley's, can have profound clinical consequences. And certainly how catastrophic failures like in Cora's heart attack highlight the absolute dependence on continuous energy flow. Understanding these fundamental processes really is key to getting a handle on both health and disease. It's a dynamic story playing out inside every one of us all the time.
Absolutely. Now, to leave you with something to think about, building on this incredible picture of adaptability and also fragility, given the remarkable redundancy and the fine-tuning built into our bioenergetic systems, what do you think might be the earliest, most subtle biochemical clue or indicator that an individual's long-term cellular energy health might be starting to decline, maybe even years before any obvious symptoms actually appear? Something to ponder. Thank you for joining us on this deep dive.